Something about myself. I work at the Institute of Marine Research in Bergen in Norway. I lead the population genetics group there. We work on everything from sea lice and copepods all the way up to marine mammals. My group works primarily with fish, however, and for much of my career I've been working on genetic interactions between farmed escaped salmon and wild fish in the rivers. I know this is a topic that creates a lot of emotion. Often it's a bit of a polarised debate where you have the wild fish interests on the one side, and on the other side you have the farming interests. But the world's never so clear as black and white, and biology isn't black and white. Biology is full of colour, and as I will try to guide us through a talk here to give some of the details, what we know about this and what we don't know. My talk is to be split up into three main sections. First I would say something about the Norwegian production, what we know about escapees, and what we know about integration. And what I mean by integration is how much genetic material from the farmed salmon has been included in the wild salmon populations. I will talk briefly about domestication, and I'll say something about what we know about the genetic differences between farmed and wild salmon and what we've got from experiments in the wild. I'll talk a little bit about mitigation efforts in Norway, what we do. And then finally, I have a few semi-philosophical thoughts, and hopefully I don't transcend the line to politics because I don't do politics. So we first get into production escapees and integration. So as most people know, Norway is the leading producer of Atlantic salmon in the world. It started when fish were first placed in cages in the latter part of 1969. The industry expanded almost exponentially, and as we'll see by the green curve here, it's topped out at around 1.3 million tonnes. It's stayed stable around 1.25, 1.3 million tonnes a year for the last 4 or 5 years, and that is because of sea lice, the issue of sea lice and influences on wild populations. The government has kind of capped it in that range at the moment. But it's not just sea lice that's important in Norwegian aquaculture, it's escapees. If we look at the figure here, we see the reported number of escapees in Norway from 2001 to 2017. These are based upon the fish farmers' reported numbers, and as we'll come into afterwards, these are slight underestimates. You see it was climbing up to nearly 1 million escapees in the worst year. To put that into perspective, the entire number of wild salmon returning from the ocean to the Norwegian coastline each year is approximately 500,000. So we're not talking about, should we say, a drop in the sea. We're talking about a very large number of escapees relative to the number of wild salmon in Norway. What do we know about escapees? My former colleague Ove Skilbrei, who is unfortunately not with us anymore, he spent much of his career, the latter part, studying the behaviour of escapees in the sea, where did they go and what sort of patterns they showed. He used a diverse range of tagging methods and telemetry, etc. And if I was going to summarise what he came up with, this is mostly what would be a summary of his work, is that most of those large number of escapees just disappear. We never see them again. Actually, we don't know what happens to the majority of them. Presumably, they disappear into the ocean. At some stage, they die, starve and perhaps die. Maybe there's some predation of them, but most of them disappear. From the genetic interaction point of view, this is kind of the good news, because if those numbers of escapees I showed on the previous slide have actually entered the rivers and tried to breed, we would be at a completely different level of genetic impact than we are today. Recaptures in sea. When they've escaped from the farms, can we use measures to recapture them back from the sea quickly? Well, it's difficult. Really, it's not possible. With the exception of large escaping fish a long way in a long Norwegian fjord where there's sports fishermen and people using nets and catching some of them, generally you can't recapture them at the site. It tends not to be effective. Especially if you go to coastal sites, the fish maybe just disappear within a few days, that may be just 50, 100,000 fish that are accidentally released. They may be away within, maybe 100 kilometres within just a few days. So they disperse quite quickly. When it comes to recaptures in rivers, what's the patterns there? Well, it's quite complicated, but if we kind of make it a bit simple, we'll say is that if fish escape in the summertime, shortly after we put them in cages as smolts or post-smolts, those fish will tend to show the normal behaviour of a wild salmon smolt. They will go to the sea, they will spend 1, 2, 3 years at sea, and then they'll come back to the rivers. Yes, most of them will die, as most of wild salmon do die on the way to the ocean and back, But when they come back, they'll have a body shape that looks similar to a wild fish, and they will be able to reproduce in the rivers. We also have the other types of escapees on the other end of the scale, where we're talking about fish escaping at, say, 3 or 4 kilograms fish in the summertime, and then if some of them are sexually mature or on the way to mature, then it's a short way from the farm to the river, and they can swim in and reproduce. But if fish escape between those 2 points, let's say as a 1 kilogram fish in the winter time, generally those are not the ones that will survive a year and come into the rivers. So it's mostly the smolt migration, should we say, escapes of smolts in the summertime, or larger fish up towards the spawning time that would be the main ones entering the rivers in Norway. From his extensive data looking at the recapture rates of farmed escaped salmon in time and space, he estimated that the official Norwegian statistics reported by the fish farmers themselves is approximately a 2 to 4 times lower estimate. So the real numbers are 2 to 4 times higher. So in 2006, the real numbers could have been perhaps several million real escapees into the sea. Okay, so that's what the escapees do, but do they turn up in the rivers? Well, in Norway, we've been monitoring the frequency of farmed escapees in a number of rivers since 1989. In 2014, the Norwegian government decided they wanted to increase, should we say, the quality and the quantity of our monitoring programme, and they started what we call the National Monitoring Programme. This is based upon multiple institutions. I led the programme in the early days just to establish it, and now my colleague Vidar Venevik is the leader of this programme. So we monitor annually farmed salmon in 200 rivers in Norway. We do this by snorkeling counts. They go in the river, they snorkel, they're experienced teams, they count the wild salmon and they count the farmed salmon. They also have the ability to remove the farmed salmon. We have sports fishing data. Sports fishing, you have to deliver a scale sample in almost all Norwegian rivers. We read the scales and say who is farmed and wild. We also have some netting and some autumn surveys done, and we have a few infrastructure facilities like this. And what this gives us is, with this entire network of monitoring, is a very good picture of where the escapees are in Norway in time and space every single year. There's a lot of money and effort that goes into this programme. The logistics are huge. This is what it looked like in 2016. So there is 200 dots in there. It doesn't look like it. Some of the dots are on top of each other. Green is where in that year we observed less than 10% escapees in the sample from that river. Yellow is where we are not sure whether there was above or below 10%. And red is where we are sure that the estimate is over 10%. For all of these rivers, we have all of the raw data presented. This report has 500, 600 sides of attachments online. You can go in and look at all of our raw data. We have stripped all of our clothes off, metaphorically speaking, and shown everything we have. This is a simplification. What you will note is fish farming is present from Buknafjord in the southwest of Norway all the way up to here. And it's only on the southern tip of Norway that you have no fish farming because it's exposed coastline. But what you'll see is that these red rivers and the yellow rivers, they are distributed along the Norwegian coast. Now, you might look at that and think that doesn't look so bad, but some of those green rivers have 5 or 6 or 7% escapees in them, and maybe the next year they have 20%. But that's how it looked like in that year. Now, if we was to look at how does it look like over time? So that's just one look, 1 year. How does it look like over time? So this red line is the trend line for the autumn surveys, and the blue line, I don't know if that's blue or green, sorry, but I can't see, but it's either blue or green, that line. This is from the sports fishing surveys. Now, automatically say, why does sports fishing give a lower estimate than in the autumn surveys? Well, typically, farmed salmon will enter rivers later than wild salmon. So if we base it on the summer fishing surveys, we get one estimate, but then we come back to the same river again and take a survey later, the estimate is higher because that's when the farm fish are starting to enter the rivers. Nevertheless, we believe the red line is an overestimate, the blue or green line is an underestimate, and the real number is somewhere between those two things. Without getting into technical detail, we tend to take the average of those. What you'll see is some periods there's been large numbers of escapees in the rivers, but there tends to be a decline downwards with time. The numbers of escapees entering Norwegian populations has decreased over time, but it's been consistently there. And the major question we had when we started to work on the genetics is, well, this is a lot. We're talking 10, 15, 20% farmed escapees on the rivers for 20, 30 years. How much gene flow, how much integration from the farmed salmon has occurred in these wild populations? So the first study we did, we've done several studies on this, and there's come a couple afterwards. We wanted to look at that. We specifically wanted to see, has there been integration of farmed salmon? It's not that long ago that we didn't know that in Norway, ironically enough. So we had an initial study, was based upon 21 populations. We took scale samples from the populations in the early or previous to farming industry. So we took samples from the 1970s and 80s from these populations, and we reconstructed the genetic architecture of those populations. Then we resampled the populations in 2010 and 11, and we created what they look like now. Then we used some fancy statistics, some fancy diagnostic genetic markers that allow us to track integration of farmed salmon into the wild population, and then we computed how much gene flow has occurred. After our studies, there's come a massive amount of data from Carlsen et al. And in this one, Disserud et al. I'm part of this. This is a collaboration by several institutes. In Norway, as of today's date, we have an estimate of integration of farmed salmon in 175 salmon populations in Norway. If we look at this pie chart, that doesn't add up to 175. This is an old pie chart, but the frequencies are the same. We say that 2/3 of Norwegian populations have a trace or a large genetic change based upon molecular markers in the population. So farmed salmon have successfully interbred at the level we can detect it genetically in 2/3 of Norwegian's populations. 1/3 we can't detect anything. In 1/4 of those populations, we detect what we classify as a large and distinct genetic change, and then we have an estimated integration level of 10% or more, which means in 1/4 of Norwegian salmon populations, we have 10% or more integration of farmed salmon. Now, this figure shows, again, this is many dots that you can't see, it shows that there's really nowhere in Norway where integration hasn't occurred to some degree, with the exception of this area around the south of Norway where there's basically no farming and they have quite robust wild populations. So the bottom line here is that there's been a lot of escapees and they have left a genetic footprint in Norwegian rivers. Well, what's the patterns of this? Is it all rivers equally affected, or how is it? Well, we don't need to get caught up in these two diagrams, but what they illustrate is there is, of course, a relationship between the frequency of escapees, how many escapees there is in the population, and how much integration occurs. That doesn't explain everything, though. More escapees, more integration, but that explains about 50% of the variation we see between rivers. So what is explaining that? Well, what we see is that we observe the highest integration, the highest gene flow levels in what we call weak wild populations. For example, the River Vosso and the River Oppo in the west of Norway, they have some of the highest integration levels. The River Oppo has approximately 47% of the genetic makeup of that river now comes from farmed salmon. Why is that? Well, that is a population that had low numbers of spawners in a period where there was lots of farmed fish coming in. The farmed fish come in, they're inferior at competing spawning. The work of Fleming et al. Has shown that farmed salmon are poor competitors on the spawning grounds, but when you come into a wild population where there's quite a lot of free spawning habitat or the competition's low, maybe they get a foothold there, they manage to spawn successfully. Whereas they come up into a robust wild population, a real fight on the spawning grounds, the farmed fish that are inferior competitors perhaps don't manage to get their foothold in there. So we see that small and weak populations in Norway, and also from the Canadian side, are those most at risk for genetic integration. We also show genetically that the integressed populations, i.e. the ones where farmed salmon have successfully interbred, become genetically more similar to each other, kind of washing out that genetic diversity among populations that existed for thousands of years. We're looking into biotic and abiotic predictors of integration. For example, just to give one example, we think that if a river has a waterfall in the lower stretches of the river, quite a tough waterfall that's tough for wild salmon to pass, this is difficult for, say, an untrained farm salmon to pass maybe, the wild fish will pass it. So that waterfall in the lower part of the river is maybe a way of kind of nature's way of protecting against a farm salmon integrating in that population. Even if lots of farm fish try to get in there, not many of them manage to go over that waterfall necessarily. Of course, this depends on the waterfall, etc. But that's just an example. So that was escapees, the patterns of escapees and integration. Now I'll move on to domestication and what we know about. Are farmed and wild salmon different? Do we really care if they interbreed with wild populations? Well, just to talk very briefly about domestication, when Norwegians started farming salmon in the early 70s, they started breeding programs immediately, which is probably part of the reason why the industry has been so successful. Norwegian salmon breeders have been very effective at breeding a fish that functions very well in the farming environments. We're now up to around 12 to 15 generations, depending on the breeding strain, and they originated from approximately 40 or more wild populations in Norway, mostly from the western side, not from the top of Norway. They've, of course, selected for traits such as growth, survival and disease resistance, delayed maturation and flesh characteristics. They now use genomic tools to make the selection. So I would say at this moment we're in a process where we're going to the new level of possibilities to select. And they've been very effective. Just about everything that we've looked at of farmed and wild salmon, they are different to each other. Behaviour experiments, physiology, gene expression, anything we've looked at have been different to some degree. However, it is growth that has shown the largest difference between them. We've done multiple experiments at our institute. We're blessed that we have an experimental fish farm of, shall we say, world-class quality. We've been breeding farmed and wild and hybrid salmon and looking at their growth rates in a million different types of experiments. And as we see up here, these are the growth of the farmed families, some variation between them. These are the hybrids and these are the wild ones. There's very big differences. Again, we look at yet another experiment, the farm, the hybrid and the wild. There's a big difference in the growth. I could carry on like this all day long. Farmed, hybrid and wild, there's differences there, no matter what. And typically, under farming conditions, farm fish will end up 2 to 4, sometimes 5 times larger than the wild fish under those identical rearing conditions. So it's clear we've changed these fish by domestication. The fish farmers and the breeders have been exceptionally good and done what they wanted to do. Okay, they're genetically different, they grow differently, but what happens when they get in the wild? Do their offspring survive? Are they able to find food in the wild? Can they survive? There is 3 empirical studies done in the wild where they've released the offspring, usually eggs, of farmed hybrid and wild salmon in the wild, or they've allowed farm fish deliberately into a river system to spawn and followed their offspring through the life cycle. I don't have time to go into details in all 3 studies, but in the Burisul, in the Irish system, these are iconic papers, these first 2, they showed that the offspring of farm salmon have a much lower survival in the wild than the offspring of wild salmon. There they used Norwegian Moe strain against the local Irish strain. In the IM system, this is in Norway, Fleming et al., they did a slightly different study. They allowed the farm fish into the river after taking a biopsy sample. They was able to spawn with the native population and they measured the survival of the offspring through the entire life cycle. I think they calculated the lifetime success, which included the lack of spawning success of the adults, to be 2%. In Goodall, we have this system in Hardanger. We've done our own studies there. I'll talk a little bit about that. In the River Goodall, we've planted out 500,000 salmon eggs of farmed, hybrid and wild from 170 pedigree-controlled families, and we've had about 10,000 recaptures. There's no normal funding system that pays for this sort of research. We've just done it, which has not always been popular, but we've done it anyway. So we've planted the eggs in there, we've counted all the eggs from the families, and we looked at various things such as age, size at smoltification, age at smoltification, survival of the families, timing of smolt migration out in the river, and so on and so forth. I won't get into the details, it's too much for now. But if I was to bring it down to a simplified slide, is that we see that the survival from egg to smolt in the river in the freshwater stage is approximately twice as high for the wild population to the domestic population in there. It varies a little bit between our cohorts, but that's roughly that. Now, what we have to consider in this study is our wild population. We didn't have a local wild population. We had to use a surrogate. So I would say that this is a kind of a minimum estimate of the differences in survival. The Fleming paper and the McGinnity et al. Papers, they have a much larger survival difference. So the bottom line there is that the offspring of farm salmon do have a much higher mortality in the wild, and hybrids are kind of somewhat intermediate. So when they interbreed with a wild salmon, they will also bring them to a lower fitness. This is a hell of a job to explain this one, and even Gaia Ballstad, the first author, has a job to explain this one without making people shudder. What I will try to simplify a little. Don't look at the figure just yet. This is the first empirical evidence of what happens in a wild population when integration of farm salmon have integressed. What I showed you just now from the Burrisul and Ims and Goodall are experiments where we've controlled it. This is the real world. So what they've done, they've gone into 62 populations in Norway where they know farm salmon have interbred. They have used genetic markers to look at every individual in that population born in the wild, and they've sorted them as pure wild genetically and farmed, or should I say hybrid, and basically whether they're genetically mixed or whether they're pure. And that's on this scale. And then they've looked at the age of maturation of the fish going to the sea and coming back again, and they've looked at the size of maturation. And without getting too much into the details, because I could spend half an hour trying to explain this, what they see is that there are differences in the age at maturation and the size at maturation in populations where farm salmon have integressed. So this, you can summarise this as, this is the first empirical evidence in the world of biological or functional changes in wild salmon populations from integration of farm salmon. This sort of data will be emerging more and more in Norway. We're working heavily with this. Over to the third main section, mitigation strategies and what we know. The first thing is the River Etna. This is a population in the Hardangerfjord. It was a population that has about 2,000 adult salmon in the run each year, give or take. It was a population that both us and the Research Institute NINA have computed is approximately 20% genetically mixed with farm salmon in the population. There's been lots of escapees and it's genetically mixed. It's the biggest population in that area, and the local river owners and the fish farmers wanted to do something, so they paid for this infrastructure to be placed. This is a ward... I can't remember the name of it, but it's a trapping facility. These are floating bars. The fish come in here, they hit that, they move here, and they go in here, and they're caught in here. Then we sample every single fish. Of course, the farm fish are killed and removed and sampled. The wild fish, we take a little sample from them, and we release them back into the river, and they're angled further upstream. None the worse for that, in a way. So this system allows us to protect the population from further integration of farm salmon. Now, for us, this is a very nice model system to work with, because we are genetically analysing all fish going into the river, which starts to be many thousand, and many of the smolts coming out, and then we have the ability to look at how natural selection will eventually clean up, or should we say purge, the genetic integration of farm salmon from this population, again with a lot of biological measurements. But what this system also gives us is a lot of, should we say, anecdotal science. And we have a couple of papers here where we've been able to look at the escapees entering the river and say something about them. In the river, we have typically in this period had 100 to 150 escapees just in that one single river. From genetic analysis, we know that escapees originate from multiple farms. It's not like just one farm one year, should we say, are the source of the escapees, then next year it's another farm. There is multiple farm sources. All of the 3 breeding lines and multiple companies are involved in escapees going into the river. 81% of those escapees in this analysis in 2014 were positive. I have to make sure I use the right terminology now. They were positive for one or more of the infective viruses that are in the farming industry. Most of the escapees entering this river system have escaped the same year they entered the river. So roughly 70 to 80% of the escapees entering that river have escaped from fish farms the same year they entered the river. whereas about 20 to 30% of them have probably escaped a smaller fish, probably a smolt, 1, 2 or 3 years earlier, gone to sea and gone back and moved into the river. We do see some immature salmon coming into the river. Well, that's a bit strange, perhaps a bit confused or whatever, or not maturing properly, but they tend to stay in the lower reaches. I won't talk about traceability much. It's something we have worked a lot on, but we have a DNA method for tracing farmed escapees back to the farms of origin when the fish farmers have lost fish and none have kind of owned up to it in a way. It's the Directorate of Fisheries in Norway that invoke use of this method, not us. We just do the genetic analysis, and usually we're able to tell where the escapees have come from. Sometimes it's a bit complicated and there's a lot of conditions of that. I won't get into that, but that's the only... We don't have any physical tagging of fish in Norway. It's been discussed, but the industry is too huge. But here they've used this method as and when, in a way. It's not the solution to everything, but it works when we need to use it. A little bit about triploids. The industry has been looking at producing triploids which are sterile as a way of stopping further genetic interactions. Very sensible. But some of the questions are, will triploid salmon just cause other problems? Will they just swim up to the rivers? Will they try to spawn with wild fish and they're kind of shooting blanks, so it's not really helping? What will happen in the rivers? Well, the first thing is that we've done a proof of concept study, and we see that triploid salmon males, sometimes they don't get sexually mature, but they produce what we call these secondary sexual characteristics, you know, this kayak, and they look like they want to spawn, but they've got nothing to spawn with. We've done an experiment and we've shown that the wild female here is spawning together with this triploid sterile male, and she is willingly dropping her eggs with them. So the proof of concept that triploid sterile fish could swim into a river and spawn with a wild fish has been demonstrated. However, we don't believe this will happen very often. The secondary sexual characteristics, maturation characteristics in farmed salmon is a rare occurrence, so we don't believe this is a major issue. However, if it's going to be a major issue, first they've got to escape from the farm and then they've got to swim to freshwater. Why do salmon swim to freshwater? That's because they're sexually mature. When you're maturing, the body says, now I need to go to freshwater to spawn. But if you haven't got the sexual maturation process going on in the body, perhaps you don't go to freshwater. We got at this by, in Norway, in the period 2007 to 14, the Norwegian aquaculture industry produced approximately 2% triploids accidentally. Without getting into details, when we take eggs out of fish and sperm out and we mix them, we produce 2% triploids in Norway accidentally. It's just a natural occurrence as well. So what we thought is then, okay, if we analyse escapees in the rivers, then we can see if the triploid escapees are just as likely to enter freshwater as a normal escapee, then we should see 2% triploid escapees among the escapees in the rivers. So we genotyped, well, 4,000 escapees over the same time period from these rivers, and the bottom line is we only saw 7 triploid escapees. So what we can say from this study is that if we used triploid salmon, not only would that stop further genetic interactions because they could not reproduce, we would also reduce by 10 times the numbers of escapees entering rivers and therefore reducing the potential for disease interactions or other ecological impacts. Of course, this is just based upon these studies. We can't generically say this would be the case for the whole of Norway. Nearly done now. In Norway, we estimate the number of escapees in rivers each year, I've already talked about, and that gets produced in this document. The Institute of Marine Research, we do an annual risk assessment of genetic interactions based upon these data, and when a river is red, there is an organisation paid for by the farming companies called URO, which is the organisation for oppdretters. Yeah, I can't remember now, sorry. Yeah, it's a mouthful for even Norwegians. But basically, they use our list of rivers here, and then they pay for a clean-up. So, for example, this group here, they will use harpoons or nets or whatever they can. They will go into the areas where they know there's farmed escapees, capture them in the rivers, and remove them out in a way. So this is a way of reducing the numbers of escapees in the rivers. And that brings me over to my last slide. I think I'm done well in time, at least reasonable. So if I'm to summarise this up, all of the empirical evidence we have indicates that integration of farmed salmon in wild populations is going to be negative. There's nothing that indicates this will be positive. In the early days, there were some people suggesting, or perhaps playing devil's advocate, saying perhaps we're supplementing with new genetic material or whatever. There's nothing that indicates that. All theoretical and empirical data suggests this will be negative. All of the evidence suggests this will be dose responsive. The more escapees you have in the rivers, the more likely you are to get integration, the more likely you are to have changes in those rivers, changes in the productivity of the population, perhaps changes in the, should we say, the phenotypic traits of the population, whether it matures like this or this, and ultimately challenging the evolutionary trajectory and well-being of these populations. But sometimes I feel like we've been working on this topic and it's a bit like climate research. In the world, most scientists working on climate, they say that it's getting warmer, and there's probably a million models out there saying it will get warmer by 2 degrees or 4 degrees or 5 degrees. And the point is that you don't know until it happens. They're just models. And I feel a bit like with genetic integration we're there, is that we know it's going to be negative, we know we're going to decrease the viability of wild populations, but by how much and on what time scale, we're into very tricky things. We're talking about evolution. Predicting global climate change is all about physics. It's actually gloriously simple. This is evolution, this is tricky, this is biology and diversity, and nature has a way of challenging integration from these farmed salmon, but when we're continually integrating these populations, maybe we're overriding nature's ability to clean up. And this brings me to my almost going into politics, but it's not, is the 3 R's. You have the 3 R's in animal welfare research, and I think here the 3 R's are reduction. We need to reduce the numbers of escapees coming from the fish farms. Of course we do. Fish farmers have no interest in losing their money. No one will want to do that, but we've got to continually strive to that. Removal of the escapees from the spawning grounds. When they have escaped, we should do what we can to get rid of them from the spawning grounds before they have a chance to interbreed. And not least of all, I think we really need to look at a reproductive barrier. I've given one example with triploidy. There is some problems with triploids, so I'm not saying that's the ultimate solution, but a reproductive barrier would stop this. And when I talk about this in Norwegian forums, I almost always end up by saying, let's kind of make fish sterile, let's rear sterile fish, then the whole field of genetic interactions just evaporates instantaneously in a way. In Norway, there's 2 things that regulate the industry. It's sea lice, which I'm sure we will find a solution for sooner or later, and there's genetic interactions. We have the solution for genetic interactions. It's very simple. If we just either stop them escaping or we make them sterile, then there's no more genetic interactions. Then half of my career can just evaporate and we can get on and do something more sensible in a way. So it's perhaps stating the obvious, but on that note, I think I'll just finish before I start getting into a real rant. Yes, thank you.